Hi Emma, what are prokaryotic cells, and how do they differ from other cells?
Prokaryotic cells, like bacteria and cyanobacteria, are unique in that they don't have membrane-bound organelles. That means there's no nucleus or other complex structures like mitochondria or the endoplasmic reticulum. They're quite small, usually between 1 to 5 micrometers in size. You'll find them in various shapes like spheres, rods, or spirals. Cyanobacteria are especially important because they produce oxygen through photosynthesis.
Do all prokaryotes have the same structure?
Generally, yes, but there are some differences. For instance, archaea, which are also prokaryotes, have a unique cell wall composition. Antibiotics that target bacterial cell walls don't work on archaea because their cell walls are made of different materials. Interestingly, archaea share some features with eukaryotes, such as the way they handle genetic information. You can often find them in extreme environments, like very hot, salty, or acidic places.
Can you tell me more about the bacterial cell wall?
Sure. The bacterial cell wall is mainly made of a substance called peptidoglycan, which is a mesh-like structure composed of sugar derivatives and amino acids. The sugars, N-acetylglucosamine, Ng, and N-acetylmuramic acid, NM, are linked together, and short peptide chains connect these sugar molecules. An enzyme called transpeptidase forms cross-links between the peptides, strengthening the wall.
How do we classify bacteria based on their cell walls?
We use a technique called Gram staining. It helps to distinguish between two major types of bacteria, gram-positive and gram-negative. Gram-positive bacteria have a thick peptidoglycan layer that retains a violet dye, so they appear purple or blue under the microscope. Gram-negative bacteria, on the other hand, have a thinner peptidoglycan layer and an additional outer membrane, which doesn't retain the violet dye. Instead, they appear red or pink after being stained with a counterstain like safranine.
What's the role of plasmids in bacteria?
Plasmids are small, circular pieces of DNA that replicate independently of the bacterial chromosome. They often carry genes that give bacteria an advantage, like antibiotic resistance, the ability to produce toxins, or fix nitrogen from the atmosphere. In genetic engineering, plasmids are used as vectors to insert specific genes into bacteria, which can then produce desired proteins.
How do bacteria move?
Many bacteria use flagella, which are long, whip-like structures that help them move. Some also have pili, which can help with movement but are mainly used for attachment to surfaces or other cells. Pili are also involved in a process called conjugation, where bacteria exchange genetic material.
What's sporulation?
Sporulation is a survival mechanism used by some bacteria. They form spores, which are highly resistant structures that can survive in harsh conditions, such as extreme heat or dryness. These spores can remain dormant for years, and when the conditions improve, they can reactivate and cause infections.
What about antibiotics? How do they work?
Antibiotics are chemicals that can kill or inhibit the growth of bacteria. They work by targeting essential bacterial processes like cell wall synthesis, protein production, or DNA replication. For example, penicillin inhibits the enzyme that forms crosslinks in the bacterial cell wall, causing the bacteria to die. However, overuse of antibiotics has led to resistance, where bacteria evolved to survive antibiotic treatment.
What happens when bacteria become resistant to antibiotics?
When bacteria become resistant, it's much harder to treat infections. Resistance can develop due to the misuse of antibiotics, like not finishing a prescribed course or using antibiotics for viral infections. Some bacteria acquire resistance through horizontal gene transfer, where they receive resistant genes from other bacteria. These resistant bacteria can spread, making infections more difficult to control.
Are there alternatives to antibiotics?
Yes. Researchers are exploring alternatives, like antimicrobial peptides, AMPs, which are natural components of the immune system. These peptides can kill bacteria by disrupting their membranes. Other options include bacteriophage therapy, which uses viruses that specifically infect and kill bacteria, and certain medicinal plants with natural antibiotic properties.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.